and get started to keep on time. So welcome. Thank you for coming. This is our second core showcase in a series that we're hoping to have several times a year, about 3 or 4 times a year, where we get to connect researchers to the various core technologies on campus and also learn about some of the new and exciting technologies that these core facilities are bringing in every year. I'm the organizer, I guess. I'm Mike Henney. I'm the director of the LMND. Just a few announcements before we kick off. So I want to start with thank yous. So big thanks to Deb Carlson and the Provost office for supporting this event, allowing us to do this. Also to our administrative staff, who I think just walked out, Kiki Fuller and Natalie Valleys for helping put this together. Thank you for all of you for coming. New thing different from the last one we had. If obviously you're welcome to take notes, but if you want to hear something again, we're actually going to record this entire session, put it on the LMND website, which you could find by just going to the cell biology homepage, the UT Southwestern cell biology homepage, and clicking on LMND. So this video and all subsequent events will also be put on that page. And also, we'll have snacks and treats outside as soon as this is over. So if you have a few extra minutes, feel free to stick around, maybe talk to our speakers today about some of the technology that they're going to talk about. So without further ado, let's jump in. I think Chad is going to be our first speaker. We're going to hear about the Macromolecular Biophysics Resource. So let me get you started. Okay.
Great. Thanks, Mike, for the introduction. Thanks also to Mike for organizing, and also to Deb Carlson, and thank you for coming out and listening to me talk today. So I'll be talking about the Macromolecular Biophysics Resource here at UT Southwestern. My name is Chad Brautigam. I direct it. So we are a core laboratory. We were formed concurrently with the Department of Biophysics in 2012. But I want to make very clear that we are not just dealing with people from the Department of Biophysics. Any person from any department here at Southwestern is welcome to come and use the core. And in fact, we have interfaced probably with every basic science department here and center here at UT Southwestern. Last count I had was something like 20. And we actually have 93 active users in iLab right now, so it's quite busy. So we have a number of instruments that can make high quality in vitro biophysical measurements on highly purified macromolecules. So I also want to emphasize that for these very precise, very high quality biophysical measurements, we are usually going to be dealing with a purified protein, purified nucleic acid, some kind of macromolecule that's been highly purified. Now there are some exceptions to that, and we can talk about that if you want, but that's sort of the general rule. The measurements that we make, I would classify as either being hydrodynamic in nature or thermodynamic in nature, and I'll get into that distinction in a moment. The instrument is housed just next door to this building over here in ND, on ND8 and ND10. And we offer experimental planning, consultation, training, and other services. And of course, there's a website. So just it's under the core type of websites here at Southwestern. We are 2 people, so we're not a big core. But I think we get a lot done for just 2 people. Like I said, 93 trainees and counting. And that's just currently in the iLab system. So myself, of course, and then my right-hand man, Scott, who is really truly excellent. All right, so here are the 2 classes of instruments that we have, and the first class I'll introduce is the hydrodynamics. So we have an analytical ultracentrifuge, dynamic light scattering instrument, size exclusion chromatography coupled to multi-angle light scattering, or SEC-MALS, and mass photometry. And I don't really have time during this 20-minute talk to go into the physical principles that are measured and the actual measurements being made by each one of these instruments. But I will tell you that in general, these are going to be instruments that measure for macromolecules, their size, their shape, a quantity known as the diffusion coefficient, or that's basically how fast they move around in solution. The molar mass, of course, quite important. And related to that would be oligomeric state, whether your, let's say, protein is a monomer, a dimer, trimer, or a mix thereof. We can deal with that also. Our thermodynamic measurements then are going to be made with these instruments. We have a microscale thermophoresis instrument, sometimes called MST, isothermal titration calorimeters, or ITCs, circular dichroism spectrometer, or a CD instrument, and then I would also classify our AUC as being able to measure some thermodynamic quantities. And those types of quantities are going to be things like melting temperature, how stable is my protein or nucleic acid, binding energetics, what's the KD? For calorimetry, what's the enthalpy? What's the entropy of the interaction between one molecule, one macromolecule and another? And in this case, for ITC, we can actually measure not only interactions between macromolecules, but even macromolecules and small molecules, or even small molecules with small molecules, because all that's measuring is heat. We also can use these to figure out the stoichiometry of interactions. And I would argue that this might be one of the most important things that we can measure. I don't think you have a handle on your model until you know its stoichiometry. So that's the instrumentation that we have on hand. Now, I'd like to go through the model of how the core works really quickly. So a prospective user would contact MBR personnel. We would then contact them back. That's usually me. We set up a meeting to discuss experimental strategy. Then we meet with the user and make suggestions. Now, I consider this maybe to be our most important function as a core. And that is because we can sit down with the user and really begin to understand what they need and how to get it. A user might come in and say, oh, I want to do an ITC experiment. And then, after we get into it, we actually might say, well, you know what, ITC might work, but I think MST's got a really good chance here. It's going to use much less material, et cetera. So this, I really feel we do a lot of this. And sometimes we tell them, you need a different instrument that's not in the core. Go use someone else's instrument. Sometimes we have to lose business, but we give them the best advice that we can. And I really think that this is super important. Most often we set up training. So this might operate a little bit differently than some cores that you're used to. And that is, we don't just take stuff and do stuff with it and give you a report. Very often what we do is we take someone from your lab and train them how to do the experiment themselves. And then they can use the equipment later at their leisure. We train the user acquires data, usually on the system of interest. We also train the user to analyze data, of course, very important. And then we can provide assistance with publication figures, verbiage, et cetera. Again, after this is done, the user is considered to be trained. And in most cases, that user can just go into our iLab calendar and reserve future time, whenever they want to, as long as, it's during core hours. Okay. Now, we do have another mode, and that mode is a service mode. We can do what I just said that we didn't do, and that is we can take your protein or nucleic acid, macromolecule, whatever, and do an experiment on it, and then we can give you a detailed report on that. This very often comes into play during, interestingly enough, review processes for papers. So a reviewer comes back at you, and it's usually reviewer 3, says, okay, you really need to do an AUC experiment. And, you've got 2 weeks to get this back to an editor. So you don't have time to learn it. You don't have the wherewithal to learn it. So you can just give us the sample. We can give you a result. excuse me, and something to put in your response, maybe even become part of the author list on the paper. So we can do that as well. Another aspect of MBR that I think for me is super important is that we want to serve as a center of innovation. We don't want to just do biophysical experiments or just teach you how to do biophysical experiments. We want to be at the cutting edge of biophysics. And so what that means is that we're coming up with new protocols and new ways to analyze data that actually end up very often in code, because I like to code in Python. So these basically, we have developed software packages. These are, this is an incomplete list now. I need to make my icons smaller because I ran out of room across here, but we publish these protocols and software packages in peer-reviewed literature, and this has been, an ongoing emphasis for us, and I think it keeps us sharp and keeps us really at the cutting edge of what you can do in solution biophysics. So to illustrate then more about what we do, I'd like to just run through a project where several things that we did enabled more research, which enabled more biophysics to be done. Okay, so this is the story of a protein called BB0383. It's part of an operon in the spirochete that causes Lyme disease, which is Borrelia burgdorferi. And this was an ongoing collaboration between me and Dr. Michael Norgaard, who is now retired and was the chair of the Department of Microbiology. Borrelia burgdorferi and many other bacteria use ABC transporters to acquire metabolites from their environment. And this is actually very important in the case of B. burgdorferi because not only is it pretty much an obligate parasite, but it actually has to live in two different environments, in a tick and in a human, or, well, actually not usually a human, a deer or a mouse. But anyway, there's this operon of proteins, and these look like the substrate binding proteins for ABC transporters. And these are basically involved in grabbing a metabolite and usually against a gradient with ATP hydrolysis, transferring that metabolite into the cytoplasm, okay, where it gets used to support the life cycle of the organism. So Ranjit Dekka decided he wanted to look at this protein, BB0383. He worked in the Norgaard lab. And so what he did is he overexpressed the protein in E. coli and purified it. Now, these proteins have a knack for having stowaway ligands in their binding sites. And so for various experiments, we want to get rid of that ligand. So what he did is he denatured the protein in urea and then renatured it. And when he did the gel filtration column, he got something that looks like this. Unfortunately, I don't have the primary data anymore, so I just simulated this with ChatGPT. But anyway, he got two peaks, and that was very unusual because it was one peak going in. And so what we did is a experiment called mass photometry. This is one of the instruments I showed you a few slides ago. This is an instrument that basically can measure the mass of a protein in solution, and it does so by looking at an interferometric image of a glass surface that you put on there as proteins in solution land on it. And that causes a scattering event that can actually be seen by the instrument. So it's single molecule contrast counting is what it is. And those contrasts can be related to the mass. The bigger the contrast, the more the mass. And so we got a profile here that said that the first peak coming off that column was exactly double the mass of the expected monomer. And then the second peak coming off was the the monomer. And as it turns out, this must have been some kind of domain swap dimer that was completely inactive to every kind of assay that we did. And so we knew that we had to focus on this stuff. This is the good refolded stuff, the second peak that was coming off of his column. So that's what he did. We were able to use this refolded material to figure out which ligand the protein was binding to. This is thymidine, the nucleoside. Okay, so there's no phosphate on here. It's just the nucleoside of thymidine. And this is an isothermal titration calorimetry experiment. So what we're doing is we're titrating thymidine into a solution of BB0383 and getting these heats of interaction. And then, as you can see, the heats actually become zero after some time. We're able to fit that and get the dissociation constant in this case. And so we checked both thymidine and adenosine, and thymidine was the big winner. Had a much better binding constant than the adenosine. And so this allowed Ranjit to get a crystal. Okay, he co-crystallized the protein with thymidine. And here's the structure. Okay, now my core doesn't do structures, so I don't want to get anybody excited about this. I did solve this structure, but that was, that's my side gig as a crystallographer. But this is the structure with thymidine bound in the middle, and that's just the electron density for it. Great. So another thing that these ABC transporters are known for is a conformational change that occurs. And this isn't BB0383, this is a different one, but it just shows you what the drasticness of some of these conformational changes. So this is an open form, and then this is with its ligand bound. So you can see that this actually quite clearly would have a significantly different hydrodynamic radius. And we can actually measure that using analytical ultracentrifugation. So we put the protein in solution with and without the ligand, and we measure the difference of those two sedimentation coefficients under a very high centrifugal field. We spin this about 50,000 RPM. And these data indicated that there was a difference of about 0.6 angstroms, which may not seem significant, but actually does indicate a domain closure there. So it looks like a duck and it quacks like a duck. So we also wanted to extract as much mechanistic information from this ITC data as we could. And so we I've been on the forefront of trying to analyze these curves to extract kinetic information. So ITC data, we often think that it's just thermodynamics, but there's kinetic information in there. And in fact, we were able to show that the big difference between thymidine and adenosine is the off rate. The off rate is actually much, much different between the two. And so the reason that they have different binding constants then boils down to the fact that adenosine comes off much faster. Probably there's more movement, more looseness in that domain closure there. And finally, with ITC, we were able to examine all of the nucleosides. And very interestingly, we got good data for all of them except for guanosine. Guanosine doesn't bind at all. Many other proteins in this family bind guanosine just fine. Okay, so this is something that we have to track down with the structure. Now, just in the last few minutes, I'd like to go through some other data from other instruments that we have. For example, these are data from our microscale thermophoresis instrument. This measures the movement of macromolecules in a thermal field. And this allowed us to get a bunch of binding constants for nucleotides that were binding to an enzyme. And basically find out that this inhibitor here actually is quite a good binder, this inhibitor called 2-chloride ADP. we were able to check the stability of mutants of that enzyme with a RCD. So this checks the foldedness of the protein and its stability, and you can see one of our mutants was not quite as stable as the others. We can also work with membrane proteins, and we can get the oligomeric state of membrane proteins in the presence of a detergent solution using our SecMols instrument that has various detectors that will detect detergent a little bit differently than it will the protein. We can deconvolute that into mass distributions that tell us how much protein is present, how much detergent is present, and what the mass is. And this particular protein ended up being a pentamer in solution. And for the last example, I will just say that we can, our mass photometer is really, really good and really, really accurate. We had two proteins here in nanodiscs. These were membrane proteins in nanodiscs, and they were supposed to actually handshake one another. And so we have nanodisc 1 and nanodisc 2, and the mixture actually has both of them free, but also the mass of the mixture is almost exactly right there. So at that point, I do want to, my time is almost up, so I just want to thank you for your attention, and here are the relevant websites, and if there's a question or two, I'd be happy to answer them.
Questions?
Yes.
Just I was curious, we are working with proteins that get post-translationally modified with single maltose, mannose, moieties. So at the moment, we're not sure, for example, if multiple moieties get modified, or the proteomics data suggests that there are many sites. So if you were to purify this protein, could a mass spectrometry give us the number of modifications and like a separation in individually modified proteins, for example, just 1, 2, 3 modifications?
I doubt that it has enough resolution to give you, sort of Dalton level readout on how many are, and maybe Andy would have more of a. Yeah. But one thing that we can do, though, is if you have a glycoprotein, we could either probably with AUC, we can tell you how much is on there if it was extensively modified. But these kind of fine level details, I think, are closer to mass spec than to mass photometry. It's good. It's quite accurate, but the resolution isn't what Andy can give you. Yeah. Yes.
With the mass spectrometry, when you were seeing these complexes, are you able to see the non-covalent interactions, or is it strictly like covalent interactions?
It is non-covalent usually is what we're looking at, but it's a great question though, because the mass photometer has to work at very low concentrations. And so if your complex is transient or is apt to dissociate at very low concentrations, we may have trouble seeing it. The operating concentration of the mass photometer is around 10 nanomolar. So
maybe to piggyback off that, can you, obviously the different instrumentation, this is going to be different for different instruments, but in a ballpark, how much protein do we generally need to be able to have for most of the assays? Getting just a few micrograms, would that be sufficient?
For a mass photometer experiment, it would be enough. For an MST experiment, you might be able to squeak by. Most of the measurements, though, are going to be either if we want to get into ITC or let's say analytical ultracentrifugation, you're looking at hundreds of micromolar to milligram, microgram to milligram quantities.
Just to follow up on that, what sort of purity do you require for something like this? And if I purify a protein, do I have to tell you what is your
Yeah, that's a great question. And for many of the experiments, for let's say, for example, for an ITC experiment, you would probably want 95% or purer material. For analytical ultracentrifugation, we can tolerate quite a bit of impurity given the new techniques that we had to analyze the data. So maybe 80% in that case. MST can be done as long as you can label the protein specifically. It can be done in a very impure solution, like serum, for example. So it just depends on the technique, but generally, if you're looking, or secmols, for example, it could also be 70 or 80% pure. So it's unfortunately it ranges from the range of like super impure to squeaky clean, and it just depends on the application.
All right. Well, how about for the sake of time, before I move on, let's thank Chad one more time. All right. And our next speaker, Dr. Andrew Lemoff at our Proteomics Core facility. Take it away.
All right, thank you. Thank you for everyone for coming and for giving me the opportunity to talk a little bit today about the Proteomics Core. Oh. Yes. Here we go. So I'll just start with the basic why do we need proteomics? So you might just think I can do RNA-seq. Why do I need to bother with proteomics? That's going to tell me everything I need to know. Well, the genome is constant pretty much, but proteome is dynamic. And the analysis of mRNA doesn't necessarily reflect the protein content in the cell. And then some samples, serum, urine, don't have an mRNA source. And so you need proteomics for those. Different tissues have different proteins. Another check in the box for proteomics there. Plus, even once you know what proteins are there and what levels are there, proteins can be modified, post-translational modifications, like you were talking about. I'm not sure if this slide is up to date. The last I checked, there's maybe 400 different types of PTMs exist. I keep hearing about more every week. Someone comes to me with their strange PTM that they want us to look for. So there's more and more that we're learning about. So in the core, we can run a lot of different types of sample. Basically, anything that you can get protein from, we can work with, whether that's cells, tissue, plasma, serum. Maybe you're doing an immunoprecipitation, purified protein, basically anywhere, even FFPE blocks we've worked with. Anything that you can extract protein from, we can do an analysis of. And so we can accept these samples. Many years ago, we used to only accept STS page gel samples. We have evolved in the last several years. We're still happy to take your gel samples, but we can also take samples in solution, on beads. If you're doing an IP, you can just give us the magnetic beads after your pull down. Don't have to worry about eluting, cleaning up. Just wash the beads, give us the beads, and we'll do the rest for you. And we have done some FFPE block slices. Those take a little bit more effort, but we can do those sorts of analyses. As for the types of analyses that we perform, sort of split it into 3 different groups. Maybe about 10% of what we do is looking at intact proteins. This may be looking at the molecular weight of an intact protein or a peptide. And then also looking at covalent interactions between proteins and small molecules. This may also involve maybe you've labeled a protein with a dye and you want to see how many dye molecules have attached to your protein, or you've done some sort of reaction and you want to see how many PTMs are on your protein. Intact mass analysis would be the way to go for that. The vast majority of what we do, though, is in this what's called shotgun proteomics, and I'll talk about this more a little bit later. This is sort of a broad category which includes protein and peptide quantitation, as well as quantitation and identification of post-translational modifications. And a subclass of these samples. We do a lot with proximity labeling or streptavidin pull downs. We do a lot of a multiplexing method called TMT, which allows us to run multiple samples together at once and to do additional sample prep on all your samples at once. For example, if you were interested in looking at phosphopeptides in your sample and you had, say, 3 different conditions, 3 replicates for each condition, instead of us doing 9 phosphopeptide enrichments and requiring a large amount of material for each sample, we can do it through this TMT method where we label the samples, mix them together. It requires less total material, and we only have to do 1 enrichment versus doing 9 separate enrichments, so less variability. The data is just generally better. This last maybe 1 to 2% of what we do, I'm not going to talk about too much, but this is essentially the mass spec equivalent of a Western blot. You have a protein of interest. You want some information about that protein, maybe absolute quantitation. We have methods to look at that. It's especially good for proteins that maybe there aren't good antibodies for. Okay. Our Proteomics Core, we are exclusively doing LC-MS, so mass spectrometry. We have, in this slide, we have 7 mass spectrometers. Each one has a separate focus. So we have a Orbitrap Eclipse shown here, I think. No, it's not in this slide. That's fine. We have the Orbitrap Eclipse, which we use nearly exclusively for the TMT analyses. We have 2 Orbitrap Fusion Lumos mass specs. The name may not mean anything to you, but this instrument is fairly sensitive for looking at protein and PTMID relative quantitation. And this is good for fairly complex samples like tissue and cell lysates. We have 2 Q-exactive HF mass specs. They do similar things as the Lumos, but they're not quite as fast, not quite as sensitive, so they're good for less complex samples, such as IPs or gel bands. We also charge less for this service. So, if you just have a single gel band and you want to know what's in it, you don't need an instrument that's going to be able to identify several thousand proteins in that sample. In fact, you probably don't want that because it'll just make the data more confusing. And then we have a couple other instruments, one for the intact mass analysis and then one for the targeted proteomics. All of these are coupled to either HPLCs or nano HPLCs. And now we have an 8th instrument. Today is our last day of the applications training. And so we're hoping within a few weeks, once we've done some more benchmarking, we'll be able to introduce this and be able to offer samples on it. It's called the Astral Zoom. It's a fancy name. It is a very fast instrument, which allows you to get a lot of protein and peptide IDs out of complex samples in much less time. As an example, a sort of a standard that we use to check our instruments is a HeLa lysate. If we inject 1 microgram of a digested HeLa lysate on our Lumos mass spec, in about a 3-hour runtime, we typically can identify about 5,000 proteins, which is good. That's a lot. This instrument with 1/5 of the material, and this is data we got this morning, with 1/5 of the material, in about 1/3 of the time, we get nearly twice as many proteins. And so this could be useful for if you have a large number of very complex samples, we can get through them quickly, get very in-depth coverage. I can see this potentially being useful for more clinical type samples where you're not necessarily dealing with cell lysates, you're dealing more likely with, say, plasma or something like that. And so having the ability to get deeper coverage of that to potentially find biomarkers is something that I'm excited about. And another nice thing about this instrument, typically for very complex samples, if you want quantitative data, you need to do this TMT. You don't need to do TMT for this, and you can still get quantitative data, and you're not limited to basically the number of labels that are available, which is 18. You can do as many samples as you want, as long as your budget allows. In the remaining time, just wanted to talk a little bit about the various services and give some examples of work that we've done, just to maybe give you ideas for potential projects that you could do. So with regards to this intact mass analysis, we can look at peptides, proteins, covalent complexes. Our current setup, we're not able to look at non-covalent complexes. For these samples, the samples must be submitted in a solution free of detergents. So no Triton, no NP40, none of that. Biological salts, buffers are fine. And typically, we need to have a few micrograms of protein in order to do this analysis. So one, this is sort of what the data might look like that we send you. And so this is showing a protein complex where they're binding a drug to it. And so we have the molecular weight of the protein here without any compound, compound bound to it. It's about, I think it says 50,414 Daltons, and it's probably good to within about 1 Dalton. So the mass accuracy is very good. And then you can see this peak here corresponds to the protein with 1 drug molecule attached, and we have a little bit here with 2, maybe a little bit here with 3. And then someone was talking or asked a question earlier about looking at post-translational modifications on a protein. Here was an example of something we did with the Tagliabracci lab, where this was sort of an unexpected result. They provided us with this SID-E protein, and we ran it by an intact mass, and we got the molecular weight of it was 104 kilodaltons, but we also saw this series of peaks that differed by roughly 129 Daltons, which you go online, you figure out, you look at your PTM table, okay, what does that correspond to? And it turns out that corresponds to glutamylation, which I think was unexpected at the time. They did some additional studies, came back with a mutant protein. It was the same protein, but requires the presence of this SID-J and CAM proteins. They made a mutation on the SID-J protein, and the polyglutamylation went away. As I mentioned earlier, sort of the vast majority of the experiments that we're doing in the Proteomics Core involve some type of shotgun proteomics, or also called bottom-up proteomics. This essentially involves taking a protein, breaking it up into smaller pieces, running the pieces by LC-MS, and then figuring out what you had originally. This method allows you to both identify and quantify proteins, mainly relative quantitation between samples, and allows you to do the same thing with post-translational modifications on a protein, as well as localize the modifications onto the protein. I like to think of this for people who are not, or maybe not fond of mass spec or just not comfortable with it. It's the mass spec equivalent of an STS page gel. Indiscriminately identifies the most abundant proteins. You don't need any prior knowledge for the protein detection, and you get information about a large number of proteins. And with this method, we can work with pretty much any type of sample, any type of complexity. So one example of a project we did a few years ago was trying to identify and localize, both identify the target of their drug and to try to localize where it was binding on their protein. And so what we did, they had 2 sets of cells. Both were treated with a drug. One drug had a biotin tag on it, synthesized on it. The other one did not. It did a streptavidin pull down on both samples. Over here on the left, there's a silver. They ran their sample by silver stain. there was this band here at about 50 kilodaltons, which was present in the sample with the tagged drug that was not present in the non-tagged drug. So they cut out that band, and we did proteomics on it. First thing was we identified the protein as tubulin, which was lucky for us because tubulin shows up really well by mass spec. So the data looked pretty good. In addition, by looking at the peptides that were identified from tubulin and comparing their relative abundance between samples, we were able to identify these peptides here, which were actually more abundant in the sample that did not have the biotinylated drug than in the sample that did. And so we were able to identify that it was this peptide, and so we were able to identify the modification on this peptide. Okay, and with regard to post-translation modifications, as I said earlier, there's a lot of them. Our analyses allow us to provide both locations and provide relative quantification of these modifications across different samples. For this type of analysis, you typically need to do some sort of enrichment, either for your protein of interest, and that could be protein purification, immunoprecipitation, running the protein on a gel and cutting out the band, or you may be interested in sort of global modification of a sample, whether it's phosphorylation or some other modification. We offer services for both of these subsets of samples. We're happy to discuss the options for that with you. Some modifications, some PTMs that we look at, and there's more. This is just some of the more common ones: phosphorylation, methylation, acetylation, ubiquitination, glycosylation, sumolation, and we get more and more every day. Here was a paper that we did relatively recently where we looked at to us at least, a relatively uncommon modification, this symmetric dimethyl arginine. So it's basically two methyl groups on an arginine side chain, but they're symmetric. With this analysis, we performed both co-IP experiments to look at what was interacting with their protein of interest, which was PRMT5. We also did a global PTM enrichment for the symmetric dimethyl arginine modification. This enrichment allowed us to identify 165 peptides with this modification, a number of these which were significantly downregulated when the protein was knocked down. Then combined with these co-IP experiments, we showed that fused was the downstream effector of PRMT5. Okay, just last couple of slides, just some logistics of Proteomics Core. So we're located on South Campus in the Y building. We have a couple of rooms for sample prep, separate room for our mass spec in Y4322. Sample drop-off is 24 hours a day, 7 days a week in Y4106. We have a refrigerator freezer. So if you like doing sample prep and over the weekend and want to drop it off at 3 a.m. on Sunday, you can. I won't be there, but you're welcome to just drop them off and let me know that you have. And of course, none of this could be done just by myself. We have a very talented group of scientists in the core, and I just want to acknowledge them. Shumei, Joe, Sungwei, and Sophie, who do the vast majority of the work in the core. And then if you have any questions about anything, I'll be around afterwards to talk, and you can also just email me either directly or at our Proteomics Core email account. All right, thank you.
Any questions? Well, I'll start. This is the newer device, the Astral.
Yes.
You saw, I think, 9,200 proteins. Is that simply a change in sensitivity, or are these proteins that generally don't fly well in LC-MS? You just have better.
It's a sensitivity and a speed issue. So our current fastest instruments can do maybe 40 scans in a second. This one can do up to 270. And so it's, for the very complex samples, it's just able to get through more scans and see the things that are maybe lower abundant that were missed. There's also a It does the data acquisition in a slightly different method, which I didn't get into just for time purposes, but it does a slightly different data acquisition as well.
Why don't we go to our final speaker? But I think, Andy, you said you'd be around a little bit afterwards too,
Yeah,
if anybody has any other questions. All right, well, let's move to our final speaker, Dan Stoddard for our Cryo-EM facility.
Thanks for finding them.
Yes, there you go.
All right. Thank you to the organizers for having all of us and for the two talks preceding. You'll see a lot of the other cores around campus tie into what we do, especially Chad's core. So I direct our Cryo-EM facility here on campus. Give you a little bit of background on what Cryo-EM is, why you may want to use it, how to take advantage of these services, where everything is, and just a little follow-up on our Cryo-EM community in Texas. And I'll try to respect everyone's time too. I'm going to keep it under 15 if I can. So Cryo-EM involves taking a purified sample in single particle analysis to get a near atomic resolution structure once your sample's been frozen in acrystalline ice. So that's the bare bones. There's usually two flavors. Typically Cryo-EM, 80% of it right now is single particle Cryo-EM. The other flavor would be Cryo-ET or electron tomography, where you can resolve unique pieces of cellular samples specifically to near atomic resolution in some cases. So the best part about this technique is the most difficult part. There's no stains or fixatives in your samples. They're very near native or in situ. The difficulty with that is our contrast is very low. Things like carbon, nitrogen, oxygen on their own don't scatter electrons as well as heavy metal stains and things like that. So a little kind of trip down memory lane. The resolution revolution happened between 2010 and 2015. It's where the technique went from affectionately being called blobology on the left-hand side of this beta gal to a near atomic resolving technique. But really since then, a lot of developments have taken place. So just in the last 10 years, our throughput's gone up nearly tenfold for single particle, even more so for things like cryo-electron tomography. But I think the biggest change I've seen is the expert core services that are available in places like UT Southwestern and elsewhere. And this really allows these services to be accessible by people coming into the field that don't necessarily want to be cryo-EM experts, right? So this is just the graph. There's the revolution, and that's the 10 years since in EMDB entries from cryo-EM. So very active field. Where can you find us? We're in 3 locations on campus, 2 on North Campus. We have our NL facility, which was this doghouse here, as we call it, is a floating building, actually. So it's on pylons that go 60 feet down to bedrock to mitigate vibrations. We also have a cellular cryo-EM facility in the NS building. It's brand new. This is more devoted to that in situ work with Cryo-ET, specifically preparing samples to go in our TEM. So I'll talk about that a little more later. And then on our South Campus, we have a very advanced TEM as well that's geared towards solving very high resolution structures, typically for single particle Cryo-EM, but moving into tomography there as well. We have modern Cryo-EM equipment. We have 2 300,000 volt TEMs, 2 200,000 volt TEMs, and we have 2 Aqualos dual beams. The Electron Microscopy Core, not cryo, also has a brand new plasma fib, which is really interesting. You should check that out as well. But I'm not going to hammer on the instrumentation. Here's our rates. Everything I say is a core service that you can hand us a sample, or we can train you to do. So in either case, you can have it the way you want, a la carte. We subsidize these rates heavily using funds from CPRIT in the state of Texas. So that way we don't have to charge the real cost of the instrumentation. We can handle BSL-2 samples, and our queue is kept pretty short. We endeavor to do this. There are national facilities, which we also encourage you to take advantage of your time there. And we work together, and you'll see how our SBL can do both of these things. The core that we primarily work with that's customer facing.
Just for clarification, those are the adjusted rates.
Those are the adjusted rates, yeah. They're going to go up a tad bit. We've kept it stable for over a year, but not by much. So you'll find that's right smack dab in the middle. And we're a little unique. We offer 24/7 support. So if you call us at 3 a.m. on a Wednesday, we'll pick up the phone and we'll get your data going again through various means. We have, like I said, our equipment is really right at the edge, but the buildings, I think, are what make the difference. So people have really were very forward thinking when they designed and built these facilities. Daniela De Castro made the first one. She did an excellent job. It's still a perfect room. So we have less than 20% humidity, great vibration mitigation. There's no EMI in the rooms, and everything sits on piezoelectric platforms. They're active. So the sensitivity when you're magnifying something, maybe a few 100,000 X, if a plane were to land at Love Field, you could technically detect that signal. So it's important to have that vibration mitigation at those resolutions. You used to have to sit at the microscopes and fill them up and remain there for days. You don't have to do that anymore. We have external liquid nitrogen tanks that feed through vacuum jacketing to all of our instrumentation. So it prevents downtime, and it's easier because now data is automated. So you can be home or wherever you want collecting your data, thousands of micrographs a day, right? You can monitor that from wherever you are. I think these are actually Jan's ribosomes from my office. But you can go to our North Campus. We have a remote facility to collect on our South Campus. So you don't even need to really go across campus. It's like virtually you're there. It helps us for troubleshooting, and it's nice if you are on North Campus, where I would say the majority of our users are. Once the data is collected, it goes to BioHPC, which is our high performance computing consortium. It's an automated transfer. You have 2 weeks to get your data, and we locally delete the data once transfer has been verified. We're collecting a few terabytes of data per day, and so our local PCs would fill up if we did not do this. And just out of curiosity, I kind of roughly calculated how many micrographs we've taken, somewhere around 30 million, and we've never lost one. So it's a fairly robust system. But if you're, on the threshold, we are very, very open to helping you in any way we can. So give us a call. If it's coming on 2 weeks, we'll hold it for you longer. We're all people, and we're here to make your lives easier, right? We do all of our scheduling and billing in iLab, so that's convenient. Like, I think the majority of core is on campus. You can also find links to our iLab on our website. And none of this really happens the way I'm dressing it up without the SBL. They are our brother core, sister core. They're the customer-facing side. They have immense structural biology experience, and they really make this run smoothly for users that are external to UT Southwestern, whom we do not train, and users that are internal. So plenty of them go through the SBL. And even if you're trained, you can still use the SBL for any type of service you want, be it data collection, experimental, design services. You can have them help reconstruct your data for you. They'll help you write your manuscript. Anything you can imagine, they're really, really good at. So kudos to the SBL. And don't go visit their poster. There's actually not a poster. That's from a different they can also, I think they've trained hundreds of users to prepare samples too for cryo-EM. So we can also train you, but the SBL has really greased that groove, and they're very good at it. So we do have prep on campus, and actually uniquely, all this prep is in very low humidity environments, which helps at cryo temperatures because you don't want icing and freezing of crystals onto your sample. The last instrumentation one would be we benchmark our instruments quite regularly to make sure they're collecting data to a publishable standard. And in our hands, that's typically 2 angstrom data or better. So what you're looking at on the right is an information limit. That's using metal atoms to just roughly calculate how much resolution you can get in an image at a given condition. And then a biological benchmark. People like biological benchmarks for a host of reasons. They're not going anywhere, and we do it quite regularly. That's apoferritin. I think that's from our Arctica, if I remember. But these are important for user confidence. If you come to us with, hey, I don't think I got the resolution I should have, or I think something's wrong, we investigate. We have a good team there for that. So we are here to listen to you as users and bring new ideas, because our default is typically to try them. This field is changing really quickly, and so we're always kind of instituting new procedures and seeing what works and what doesn't, right? Overview of a typical, in quotation marks, single particle project. Most of it's sample optimization. So you would come with a sample to either us or the SBL and take initial data screening to feedback into what you need to do biochemically with your sample. You might go see Chad and say, what do I even have in my tube, or Andy, right? And then you further optimize that. The real culmination of this is only 1 or 2 days of data collection, and now very quick processing that could be in real time. So that's changed quite a bit, just based on throughput. And then you go back typically, as standards rise, so do publication thresholds. So you may need different kinetic states or different control structures, or you may need a different drug bound state, something like that. So that's quite a typical SBL project, or sorry, SBA project. So though they all do differ, they're individual. I mentioned cryotomography. Most of our cellular samples, in fact, all of them, are usually too thick to look through if it's a eukaryotic cell on these TEMs. You need to be on the order of a few hundred nanometers in thinness to resolve these samples. So to get our cells thin enough, you can use a cryo-focused ion beam after you vitrify or plunge freeze your cells. And that just takes something the thickness of just relatively like this room to my hand, right? That's the scale. Well, usually I'm in a smaller room when I make this analogy, but usually something like that on that order. And then you put that in the TEM and you do tomography, just like computed tomography, where you're putting a patient in a donut and tilting the machine around. We tilt the patient instead, right? Because we can't move the microscopes. If your sample is a bit thicker, we can do high pressure freezing. It takes advantage of very high, 30,000 PSI or more in some cases, to keep your sample from growing ice crystals when you freeze it, right? But this just changes that kind of room to hand analogy into an even more difficult to solve problem of targeting. What you're cutting away and what you're left with. What you're left with is what you're going to image. And if you cut away what you care about, you can't image it. So we have a really nice confocal that's integrated into this core service to get good Z stacks, but this is still an outstanding kind of challenge in the field that's not anywhere near solved. It's a case-by-case basis. It can be very difficult. So that's kind of the edge right now. We can provide just regular correlative light and electron microscopy imaging. If you have a fluorophore, this actin here you see. We can do it post-cryofib if you have an experimental sample. So for these heat shock proteins here. And I misled you a little bit earlier. I said that there were two flavors of cryo-EM. There's kind of a quasi-third called microcrystal electron diffraction. And we have this pretty new. We have a core service for this as well. It's a lot like standard X-ray crystallography, except we're in a microscope using electrons on very, very small crystals. So submicron sized crystals are, it was billed as an advantage of the technique, but really you have to have submicron sized crystals to use this. So crushing them up but Joyce Fung at the SBL does a great job running that service. And we're still kind of nascent in the post-processing, but we have a lot of great people on campus kind of generating new programs to kind of do this processing with to get to structures faster. We haven't had as much usage on that side, but this is really good for like natural products and small molecules and things like that. Not so much proteins. We can train you, like I said, to do any or all of these techniques, but it's relatively costly. If you're going to do a long project, we'd suggest you be trained. If you want to have a future doing this and maybe your future lab, definitely take advantage of the training. And just because you're trained does not mean you can't go through the SBL anytime you want for any reason also. So that's our core, a little bit about just the cryo-EM community. It's still kind of a new technology. We've only trained about 200 users since our inception. We have anywhere from 50 to 80, I'd say, active users from a lot of departments, though. So it's a really kind of widespread technology that's very interdisciplinary, and we're really just kind of scratching the surface of working together with various departments here to do complex projects. This is our biophysics retreat. There's a great group of folks doing cryo-EM in biophysics. I would like to plug, we do have a cryo-EM symposium that started last year. The second iteration of that is going to be in May in Austin. So we just got our website up. It's pretty ugly, but the conference is all ready to go. It's completely free. So as long as you can get there, it's a 2-day event, fully catered, bring your lab, the more the merrier in that context. We have a consortium for sharing time on our instrumentation. These things go down frequently, and it's nice to have time blocked at sister institutions that have similar equipment or sometimes different equipment. It's just nice to have access to maybe a newer version of something or something with a different bell or whistle that you could make use of. And it brings the community together. So Just want to highlight some of the work done by our faculty. Lots of publications from Cryo-EM since 2017, high-profile publications, and it's enabled a lot of research funding, which we were very grateful for, to continue to drive projects here in structural biology. And so just another overview of some of our core services. Single particle cryo-EM would be your big one, on, to solve a single complex to high resolution. Cryo-electron tomography, that micro-ED we talked about. Sometimes you don't need 3D information. You just want 2D electron micrographs in cryo. We can do that. High pressure freezing. We train for everything. It's the only one without an acronym, so I gave it one, lest it be left out. And then data processing services through the SBL. And they also do a lot of workshops. So how to process your data once you get it. We run these quarterly, and the categories rotate. So it's whatever the community feels we need. Actually, Yang Li back there ran the last one. I think him and Yan Han. On time, great. So with that, I'd like to thank all of our staff and the Cryo-EM facility and our SBL staff. We really do work together, and it's a group effort. So I'll take any questions.
Question. I have a nice one. What is the current size resolution? How small can a protein be? Do you think it's a good candidate for cryo?
Depends on the protein. If it's featureless and small, once you start getting too far below about 100 kilodaltons, it's very difficult to see. There are some bigger things, even that if very featureless, can be difficult to solve, especially if they're labile. It's really case by case. But you can have proteins that sit in membranes, and you can use that to reduce the degrees of freedom for your averaging. And I mean, you can get very small proteins. Things like hemoglobin. If you have a priori information, it really helps. Symmetric things really, really help. We tried to solve an RNA on its own, and it was a little too labile, and it was only about 18 kilodaltons, so very small. I think that can be done, but with a lot of data and a lot of massaging at very high res. So it's a case-by-case basis. That would be an expensive project. If it wasn't a kind of a benchmarking thing, I wouldn't do it. Yeah.
Well, if there's no more questions, we're ending on time. Thank you again. Let's thank all the speakers one more time. And we should have some snacks outside. Folks want to hang around and maybe talk to our speakers. Thank you very much. Have a great day.